for what will be an exciting presentation. My name is Robert Ninnis and I'm the Automation Business Manager for Trajan Scientific and Medical in ANZ. And I'll be your host for today's webinar. So before I introduce our fantastic speaker, I just wanted to go over some housekeeping points for today's presentation. Just to make sure that everyone is aware, today's presentation is being recorded. And if you're uncomfortable with being part of that, we understand. We will be sending the replay out to all that have registered. So you're welcome to watch the recording and join us there afterwards. We will be running a live Q&A at the end of the webinar and at points during the webinar. For all those burning questions, could you please use the raise a hand feature, which is at the bottom of your Zoom screen. When you click the raise your hand feature, I will see it and I will give the attendee the option to unmute themselves and address their questions directly to Sheena and myself. We've also enabled the ask a question feature. Now this is also located at the bottom of the Zoom window, and it's a fantastic feature for using the option of putting in questions during and after the presentation. So this feature, there is a Q&A symbol at the bottom of the screen. Click that to open the tab and just pop your questions in there and we'll answer them during and at the end of the live Q&A. Please also feel free to upvote questions if great minds think alike. You will notice there's also a chat feature, and this is if you need to communicate with Sheena and myself during the presentations. For example, if there are any potential sound or image issues, let us know, or if you just want to communicate with other attendees. And if you've missed anything or want to share the presentation with colleagues who didn't make it, don't worry, we'll be sending around the recording on demand after when it's available. So with that all being said, please it is my great pleasure to introduce today's speaker, Professor Sheena D'Arcy from the University of Texas. Originally from Australia, completing a Bachelor of Science with honours at the University of Sydney, Sheena then took flight to the UK where she undertook her PhD studies at the University of Cambridge. It was there under the supervision of Professor Sir Thomas Blundell that Sheena developed herself into a first-class X-ray crystallographer, and her passion for structural biology flourished. After completing her PhD, Sheena took up a research position at the Howard Hughes Medical Institute in Colorado, investigating chaperone function in chromatin dynamics. With a strong background in advanced structural techniques, it was here that Sheena's research started to deep dive into the structure and functional relationships of proteins and their complexes. In 2015, the D'Arcy Lab was established at the University of Texas, Dallas. Now boasting 12 members strong, their focus was on understanding the regulation of chromatin structure through the concerted action of post-translational modification, histone chaperones, and other nucleosome binding partners. Sheena and her group have made significant contributions to expanding the applications and utility of HDX. And in today's talk, titled HDX Beyond the Integrated Structural Approach, we're going to learn how it can be applied to provide structural and functional insights. Thank you, Sheena.
Thanks, Robert. So as Robert said, today I'm going to talk about how my group has been using hydrogen-deuterium exchange, and I'm going to focus on three very specific applications with the goal of demonstrating really how HDX can be applied to a diverse number of systems. Now, I came to HDX as a structural biologist. Okay, this is unlike many in the HDX field that come as mass spectrometrists. For me, I was wanting to get, the high resolution structure of my complex of interest, but it wasn't working. So it was, what is wrong? What can we learn about this complex? And that's where I first was introduced to HDX. Could we figure out the interfaces? Could we optimize our constructs? These are all things that you can do using HDX. However, over the last 5 years, as now my lab has its own HDX system, we have really come to appreciate that while HDX is really, really useful in the integrated structural approach, what it does is give the integrated structural approach a whole extra element. And that's what I've attempted to depict here with this cartoon, by instead of just looking at our complexes as static, sort of as snapshots like we often see in X-ray crystal structures, we now try to visualize our proteins in the movie form. We can use HDX to understand their dynamics. And by dynamics, I use this term rather loosely. I'm talking about the stability of a protein's conformation. I'm talking about how that protein might move in solution as particular variables are changed. And so while I'm only going to sort of touch on one story that really highlights the importance of dynamics today, this is something that I think HDX is very unique in providing this type of information. So now that I've sung the praises of HDX, I should probably tell you all what it is. So in my lab, we do hydrogen-deuterium exchange coupled to mass spectrometry, and this is probably the most common type of HDX that's performed today. And what it does is it measures how many hydrogen atoms in the protein backbone exchange with deuterium, and the deuterium is in the solvent. So you replace your water in your buffers with deuterium. The key word here is backbone. So we're focused on the backbone amide groups of our protein. So there are other hydrogens, of course, in our proteins, but we are not typically measuring these in a hydrogen-deuterium exchange experiment. This is because they either don't exchange with deuterium or because they back exchange really, really quickly, so we cannot monitor them. But the power of the backbone is that every single amino acid in your protein, except for proline, is going to have a backbone amide. So we're getting a lot of information about, in a lot of cases, almost our entire protein sequence. So why do we care how many hydrogens are incorporated? Well, we care because it tells us about the hydrogen bonding status of these backbone amides, as well as their solvent accessibility. And these are very, very important features as they relate to protein secondary, tertiary, and quaternary structure. Now, of course, this very information-rich approach does come with some cost, and that is that the incorporation of deuterium is also highly dependent on the pH and the temperature. The good thing is that there are many systems available now, and this is sort of well characterized so that we know how to control these two parameters in our experiment. But you have to be a very good analytical chemist and make sure you pay attention to these two parameters when you're doing an experiment. Now, over here, I'm showing you the exchange rate of deuterium into the backbone amides, as well as out of the backbone amides. And what's key over here is that both of them are minimized at a pH of 2.5. Now, this is very important when you're doing an HDX experiment, as we like to do time courses. So we expose the protein to deuterium for a set amount of time, and we try to minimize or quench that exchange by taking advantage of this minima and reducing the pH to 2.5, and then doing all of the rest of our sample processing at pH 2.5. Now, the other thing we do is we reduce the temperature. So we try to keep the temperature as low as possible. And this prevents this back exchange happening, which is when we lose the deuterium that's already been incorporated into our protein as we're processing the sample. So what does a typical experiment look like in my lab? Well, a lot of the experiments we do, and all of the systems I'm going to present today, are bottom-up experiments, which means we're looking at the peptide level, and they're also comparative experiments. Now, comparative experiments are amongst the most powerful in HDX. And what I mean by comparative is that you take a protein in at least two different conditions, here indicated by condition A, condition B. So what is different? Condition A may be the protein on its own, by condition B would be the protein with a ligand. So now you can see how the ligand changes the protein by comparing the deuterium uptake in these two conditions. Now, the ligand could be another protein, it could be an antibody, it could be a nucleic acid, a lipid, a small molecule, right? The options are really endless, okay? It could also be that A and B represent different protein states. So for example, it could be a post-translational modification, a different buffer condition, introducing a point mutation, or even an intermediate in a folding or unfolding pathway. But we can compare the deuterium uptake between these two conditions. So a typical workflow is shown here. And I could probably give, a 2-hour seminar on each of these steps, but just to go through it quite quickly so you can appreciate some of the data I'm going to show. The first step is that we have to take our sample in our different conditions and we have to expose it to deuterium. We then stop the deuterium incorporation at different time points by reducing the pH and the temperature. We also unfold the protein. The reason we do that is that the next step involves protease digestion. So we chop our protein up into peptides. Now I want to reinforce that this is happening after we've done the exchange. So the exchange is done in a native intact protein, but after we've quenched that exchange, we then chop it up. And the reason we want to do this is that this increases our resolution. It enables us to map where in the protein changes are happening between our conditions A and B. The peptides are then separated by liquid chromatography before being injected into the mass spectrometer, where of course we can calculate how much deuterium is incorporated because it's heavier than hydrogen. So what do the data actually look like? Well, you might see something like shown on the left here. This is a coverage map. So this is the protein sequence where underneath it, these gray rectangles indicate the peptides that we recovered from the protein. So you can see we recover quite a lot of peptides in this specific instance, and these peptides are overlapping. When these overlapping peptides give us the same result, which they most often do, it gives us a lot of confidence in the data. And when they're overlapping, it also allows us to narrow down which parts of the protein are experiencing change. The actual spectra that we collect in the mass spectrometer might look something like this. So this is the spectra for, say, one example peptide. So here we would have our spectra at our T equals 0, so our completely water condition. And then over time, you can see our spectra are shifting to the right. They're getting larger as more deuterium is being incorporated into the peptide. So the data analysis involves, identifying these spectra, centroiding them down to a single mass, and then calculating the difference in mass between each of these time points and the control, the water-only control at the top. We can then graph this on what we call an uptake plot, where on our x-axis we have time, and on our y-axis we have the relative deuterium level. So for each one of these rectangles, each one of these peptides over here, we will have one of these uptake plots. And for every condition, say A and B, we would have one trace. So we would have one trace for A and one trace for B. So you can see that this can result in quite a lot of data. So here's an example. I think this is about 1/3 of the peptides from a particular experiment. We have the green condition and the blue condition. And we're saying in which condition, in which parts of the protein, does our condition cause a change? And you can straight away identify here some parts of the protein where the blue and the green traces do not overlay. Now, I'm not going to talk about it today, but we can also just look at a single trace and comment on sort of the stability or the secondary structure of that part of the protein. But like I said, that's not what I'm going to focus on today. I'm going to focus more on these comparative experiments where we're looking at how the deuterium uptake changes between different conditions. Now, once we go from these uptake plots to sort of a figure in a paper, that's where everybody sort of has their own style. And for me, it really just depends on the specific result that we get. But I will just say my lab did come up with sort of this little application that really helps create two common ways to represent the data. One way is a chiclet plot. So this is a heat map showing the difference between two conditions. And the other way is a volcano plot, where here we're explicitly saying how big was the change and is it statistically significant? And you're going to see these two types of representations during my presentation. So this is an overview of my lab. We have a strong interest in chromatin regulation, where we do a lot of crystallography, cryo-EM, and biochemistry. But of course, today I'm going to focus on the second circle, which is the expansion of the HDX toolbox. And I have three short stories that I wish to tell you. One, which is about drug binding to a protein. The second one is epitope mapping, which we did very rapidly for COVID-19. And the last one is looking at complexes involving nucleic acids. So the first story is looking at a protein and using HDX to identify the binding sites of small molecule fragments. And this is work done by a PhD student, Darby Ball, in collaboration with the Hydrogen lab at the University of Cambridge. So the protein of interest here is CK2 alpha. The structure is shown here on the right. CK2 alpha is a serine threonine kinase that's clinically very interesting as a target for cancer therapeutics. It's a very promiscuous kinase. It has greater than 300 known substrates and is typically involved in the amplification of kinase cascades. There is already a clinical candidate in trials, this CX4945. Now, this is a pretty standard kinase inhibitor in that it's a competitive inhibitor that binds here in the ATP pocket. Now, while these can be useful, they also come with some problems, particularly off-target effects. And this inhibitor is already known to inhibit at least 10 other kinases. The goal of the project was thus to develop allosteric inhibitors. And by allosteric inhibitors, I mean non-competitive inhibitors that are binding somewhere else on the kinase outside of the ATP pocket. And this other pocket will typically be less conserved, amongst all of the other kinase family, leading to a reduction in these sort of off-target effects. Now, one strategy to do this is use a fragment-based approach. So where you start with really small molecules, figure out where they bind, and then link them together. Now, this was successfully done by the Hydrogen lab. This is the drug molecule that resulted here. And this drug molecule bound in both the ATP pocket as well as this alpha D pocket. And it's this alpha D pocket which was responsible for the specificity to CK2 alpha and sort of makes it more of an allosteric inhibitor. More recently, however, this group of labs published another series of inhibitors that they also claimed to be allosteric. And they proposed that they bound in this site A. However, there were no crystal structures available in their publications. Now, this caused us to kind of raise one eyebrow as the inhibitors really looked like ATP competitors. So we weren't convinced that they really were allosteric. So we thought maybe we could use HDX to determine their binding site. And this would kind of serve two purposes, because then we would validate the use of HDX for fragment screening or future fragment screening for CK2 alpha. So what did we actually do? We did a series of HDX experiments where we looked at CK2 alpha alone, and we compared its exchange to CK2 alpha with a fragment. Now, rather than just doing the fragment of interest, so that's this one highlighted here in yellow, this molecule 1, we did 3 other molecules. And the idea was, or the motivation was, that we were going to get fingerprints of CK2 alpha that could be used to identify future binders. So for example, molecule 6 here is known to bind in both the ATP pocket and the alpha D site. Molecule 3 binds in only the ATP pocket and just a part of the ATP pocket, while molecule 4 binds in just the alpha D site. We also did the ATP analog, AMP, PMP, as well as ADP, so we could really understand how the binding of these molecules would look in the HDX experiment. The other reason I have this table with all these numbers here is because it highlights two of the challenges that were easily overcome in this experiment. The first was we discovered that we needed to have the compounds at quite high concentration during labeling. And this was because some of them had fairly moderate affinities. And we always want to make sure we maximize our percentage bound. Now, this led to the second problem, which is that many of these compounds were not soluble at these high concentrations. So we got creative and we ended up adding some of our acetonitrile and DMSO to our buffers, of course, always doing our CK2 alpha alone in exactly the same buffer. So this insolubility and this weak binding, which we at first thought were going to be problematic, actually turned out to be quite easy to fix. So what do the data look like? So this is the totality of the data here. Each of these heat maps represents the data from one of the molecules binding. On our x-axis, we have our time. You can see we started with quite a large time range from 10 to the 1 to 10 to the 5 seconds. While our y-axis here is a list of 192 redundant peptides that are ordered from the N to the C terminus. The color on these heat maps here represents the difference between CK2 alone and CK2 with the ligand expressed in Daltons. Now, the first thing we noticed when we saw all of these heat maps was that we had this kind of unusual and recurrent protection in this region of the protein at this very late time point, with all of our molecules. Now, this told us that this was pretty much just stabilization, general stabilization of the protein due to ligand binding, but not really indicative of any specific binding effects. This meant we could easily zoom in and focus on the other areas of interest, specifically residues 39 to 55. And this corresponds to the P loop that interacts with the phosphate groups of the ATP, as well as 121 to 131, which corresponds to that alpha D site. So now we can sort of pull out the relevant parts of the data and create a much more concise and informative figure here, where we're focusing on the P loop and the alpha D site with the different molecules. And we absolutely were able to validate the system. As you can see, ADP and AMP, they have very similar patterns in both the P loop and the alpha D site, with the larger protection here for AMP, PMP, indicative of a tighter affinity compared to the ADP. Our molecule 3, it binds only in the ATP site with a slightly different pattern. As we know, it binds sort of just on one side of the ATP pocket. And then molecule 4 and 6, we see protection in the P loop and deprotection indicated by this red here in the alpha D site. Now, this is really interesting because I think it demonstrates that, binding isn't always indicated by protection or by less deuterium uptake, but this was also consistent with crystal structures that show that these molecules here kind of push this alpha D helix out, allowing it to uptake more of the deuterium from the solvent. So nothing surprising and really validating what we already knew about the binding of these molecules. So the big question was, what happened when we add the molecule of interest? So these are the data with the molecule of interest here. And I think it's very, very clear that the pattern we see here really corresponds almost exactly to what we see with our ATP analogs, really indicating that this molecule does bind in the ATP pocket and is most likely to be a competitive inhibitor. We can also go to that site A that was proposed in the previous paper. We have 8 peptides covering that region, and we see absolutely no change in them. So it would just appear like this white box here. So HDX is absolutely able to identify that this compound here was not binding at the allosteric site that they proposed in the paper. We subsequently were able to get a crystal structure. So this is the crystal structure, and we can see that molecule sitting in that ATP pocket. I've also projected one of the time points of the HDX data onto the structures here. The crystallography alone would not have been enough. However, as there was some protein engineering required, and of course we also have the role of crystal contacts that can influence exactly where small molecules bind, but the combination of the crystallography with the HDX definitely led to a firm conclusion. So we can see that the molecule in question definitely bound in the ATP pocket. It was not an allosteric inhibitor. We can see that HDX can be used to identify weak binding of small fragments. In fact, I would say it's a great technique for drug screening because you can actually see where the drug binds at a very early stage. And we also validated HDX as a useful approach in screening of fragment libraries for CK2 alpha. So that's the end of my first application. I would love to have a dialogue if people had any specific questions generally about HDX or the CK2 alpha story right now. So I will open it up for questions.
So you can do questions by raising your hands, or I'm happy to read them out if you'd like to put them in the Q&A section. Perhaps maybe to start with, Sheena, maybe I'll ask you to comment on the applications of high throughput screening with this method.
Yes, so I have not used the HDX for high throughput screening, but plenty of others have. And I think one of the things that's really common is you basically screen groups of molecules at a time. And then you can focus on the peptides, like if you have a specific pocket you want to target. If you see a change, you can then take that group of molecules and then do further screening with them. So I have not done that. I know that others are definitely doing that right now.
Excellent. I think we have one question. I'll just allow you to talk from Thomas Neville.
Hello, hi Sheena, it's Tom Neville here, work at CSIRO. I've got a question about, you mentioned the software that you created, the HD Explosion. I was wondering if that is open access, if that's available for everyone. And I also know that the Pasteur Institute, for example, they also, they've got open access software, I think called MEM HDX. I was wondering if you've tried that one and what were your experience with that was.
Yes, absolutely. So I want to maybe just clarify there that the application that we wrote does not actually do any spectral assignment. What it does is it will take your already assigned spectra and centroids, and it will just apply some statistics and visualize them, and you can customize the pictures. So it's really at that later stage. Now, as for software, there is quite a big discussion about software in HDX. MEM HDX is one that came out recently, and I will say a lot of people in the field really like it because we're really moving to trying to apply more statistical analysis to the data, and MEM HDX enables you to do that. So I think that when you first start, you use what software is available and most familiar, but then as you get further into it, you may find yourself trying other softwares and comparing them further down the line, depending on what your specific application is. I hope that was useful.
Yes, thanks.
I have to say, I do like the name of your software, Sheena. It's very creative.
Thank you. Of course, we had to brainstorm something that would be catchy. But yeah, it's really just for a basic p-value test and visualizing. Because you have such large data sets, it can be good to use software just to create different types of figures really quickly. Okay, if there are no other questions, I'll move on to the second application. And this one is going to talk about epitope mapping, basically where we're looking at a protein alone and a protein with its antibody. Now, this application is slightly different from a typical protein-protein interaction because here we're not always interested in getting the peptides back from the antibody. If you do a protein-protein interaction, you often want to get peptides from both proteins and map the interface and then potentially model it. But here we're not necessarily that interested in the antibody, so it makes it a little bit easier. But then also antibodies have these disulfide bonds, and so you have this added step of making sure you really reduce those disulfide bonds before you put your sample in the mass spectrometer. But again, many have come before us, and there are many protocols and papers available to guide you through doing this. So this is work that we did with the Maynard Lab at UT Austin. And I want to point out this was done very, very quickly by a first-year student in my lab. The reason I want to point that out is because often professors are worried that it's going to take students a long time to learn how to do an HDX experiment. This student had no prior experience with HDX or mass spectrometry, and I think in the space of about 4 to 5 months was able to produce this really, really nice data. I will say there was a bit of pressure because we were epitope mapping the coronavirus spike protein. So pathogenic coronaviruses is something which I'm sure all of us have heard of because we're in the middle of a SARS-2 coronavirus outbreak. But there are other coronavirus and there have been other outbreaks as well. For example, there was an outbreak of SARS-1 in 2002 and MERS in 2012. So the goal here was to see if we could identify pan-coronavirus epitopes that would act on all of the different coronaviruses that are pathogenic to humans. Now, in order to do this, we targeted the spike protein because all of these coronaviruses use the spike protein to mediate fusion with their target cell. So the spike protein is shown here in panel A. It is a trimer, a homotrimer. It has an S1 and an S2 domain, and the S1 domain is cleaved from the S2 domain following the S1's domain interaction with the ACE2 receptor. Now, once this cleavage occurs, the S2 domain then undergoes a conformational change and then mediates fusion with the target cell. Now, all of the coronaviruses have this spike protein. And so our question was to identify epitopes specifically in the S2 domain rather than in the S1 domain. So most of the neutralizing antibodies that you're probably reading about in the media and in the literature, they really focus on this S1 domain and specifically this this receptor binding domain that mediates this interaction with the ACE2. But we were focusing on the S2 domain because it is more conserved between the various coronaviruses. So our collaborators had immunized mice with a stabilized MERS2 S2 protein. Okay, so this was not even the coronavirus, it was not even the spike protein from the SARS-1 which causes the coronavirus. The resulting antibodies were then characterized, and 3 of them were identified to bind all 3 spike proteins. So the spike protein from SARS-1, SARS-2, and MERS. Of these 3 antibodies, 1 antibody then showed neutralizing activity in cell fusion and pseudovirus infection assays. So in vitro assays, this antibody showed a lot of promise. We also showed that the antibody was conformationally sensitive. In other words, if we denatured the spike protein, it would no longer recognize it. But then we got stuck because even with one of the world's experts on cryo-EM of the spike protein, we were unable to get a structure of the 3A3 antibody bound to the spike protein. So the question was, can we identify the epitopes specifically of 3A3 using hydrogen-deuterium exchange, and could we do it very, very rapidly? So this is the coverage map that we obtained for the spike protein. The goal was to do the spike protein alone and then compare it to the spike protein with the antibody. Now, for anybody that already has HDX experience, you might look at this and think this is a horrible coverage map. The reason I say that is you can see we have large regions of the spike protein for which we don't obtain any peptides. Now, the reason for this is that the spike protein is very heavily glycosylated. The glycosylation sites are shaded in gray here. And we did not allow glycosylation as a modification when we searched for the peptides. Now, the reason that we did this is because we had some data that showed that the 3A3 antibody had the same affinity if the spike protein was glycosylated or not. And we wanted to work quickly, right? We wanted to really focus down on as few peptides as possible to figure out what the epitope was. So we went forward. Now, these are the data from the non-neutralizing antibodies, the 4A5 and the 4H2, and we were unable to identify their epitopes. So this tells us that their epitopes are probably in a region where there is a glycosylation, but of course we were not so interested in identifying their epitopes as they were not neutralizing. But just to take a step back, these are those volcano plots that I mentioned. So on our x-axis, we have our delta change in HDX. So the spike protein alone, uptake minus the spike protein with the antibody. And then on our y-axis here, we have our p-value. So for all our experiments, we do 3 or 4 replicates, and that enables us to determine a 95% confidence interval. So if there was something that had a significant change, we would be seeing dots here. And here we have the uptake plots. And again, you can see that in all conditions, the amount of deuterium uptake is the same. So we were unable to identify their epitopes. But of course, I wouldn't be telling the story if that was the case for the 3A3. So here are the data for the 3A3 antibody. We actually had an IgG as well as a Fab. And so in panel A, we're showing the data for the IgG, and in panel B, we're showing the data for the Fab. And you can see it, I just chose one time point to show here the 100-second time point, that we're getting a collection of overlapping peptides all in this significant region. And then if we look at the uptake plots, you can clearly see a difference between the black, which is the spike protein alone, and the orange and blue, which is with the IgG and the Fab. So now if we map this onto the spike protein structure, so this is the structure of the monomer. In blue, I'm showing the data for IgG. In orange, I'm showing the data for the Fab. They're essentially identical, but you can see we clearly localize the binding or the epitope to the apex of this S2 domain. If we look at the sequence of this epitope between 7 different coronaviruses, we can see that it's highly conserved. And if we look at the structures of the spike proteins from these coronaviruses, we can also see that it's structurally very conserved. So we were able to very rapidly identify the epitope of this 3A3 IgG. Now, what's interesting is if we look at this HDX data in the context of the spike protein in its trimeric form, here again, I'm projecting the HDX data, but in gray, I'm showing regions of the protein for which we had no coverage, right? Remember, that was quite a lot because we didn't allow for glycosylations. We can see that in the trimer, when it's in this closed state, so that's when the receptor binding domains are all in the down state, we cannot have access to our epitope. And as we progressively move these receptor binding domains to their up position, these are different cryo-EM structures that are available. Now we can see our epitope is becoming more available. Before finally in this last structure here, we actually have the ectodomain of the ACE2. We can almost completely see our epitope here. So this tells us that this 3A3 epitope is conformationally sensitive and perhaps explains why we were not having much luck with the cryo-EM. So if we put this in the context of this conformational change of this S2 domain, you know, we start with the S1 and S2 as a single protein. It's the S1 that's going to interact with the AC2 receptor, leading to the cleavage of S1 from S2. Here, that would lead to an exposure of our epitope in this prefusion, as it's described, conformation, which would otherwise subsequently undergo this conformational change. This would be our epitope here, which would now no longer be represented by, recognized by our antibody. So we really have identified, you know, a potential epitope here that can be used for a pan-coronavirus therapy. So the S2 apex may be a vulnerable site for pan-coronavirus neutralizing antibodies, but I guess more generally, HDX can be used to rapidly screen for antibody epitopes. Again, before I jump into my next application, I'd love to open to the floor for questions. I see one, I think that was left over from last time, which is, can you comment on the utility of ion mobility to HDX experiments? Sure. So we have a mass spectrometer that does do ion mobility. So we do use it when we are searching our data. I have not done sort of a deep dive analysis, if we didn't use it, how that would affect our sort of coverage of the protein. But I can tell you there is a paper out there that did do this. And I believe the conclusion was that it's really quite advantageous if you have multiple charge states. So it might not drastically increase your coverage, but it will allow you to detect multiple charge states of the same peptide. We do use it routinely. Prior to starting my own lab, I had never used it, and I still had sufficient peptide coverage of my proteins of interest.
That was a great question. It's amazing how fast or rapidly you were able to really jump on this analysis. It really shows the flexibility of HDX. I think Tom's got another question. So Tom, yes. Go ahead, mate.
Hi, Sheena. Yes, I have a question around, you know, like pesky glycoproteins, how to deal with the glycosylation. What kind of like strategies are there to kind of like, you know, dig a little bit deeper? I mean, yep. There's obviously, you could, you know, things that I can think of is maybe PNGSF treat the protein before the experiment, or maybe express the protein in the presence of an inhibitor like kifonensin, or can you use different, are there different enzymes that you can use to kind of like give you different cleavage? Like, what's your experience there? What's your advice?
Yeah, so this is absolutely something that we're diving into right now, as more and more of our proteins are coming from baculovirus. So I will say, just in regards to the spike protein, the glycosylations are really quite they're mapped out, right? There's been quite a few high-profile papers where we kind of know what they are, and there is another HDX paper using the spike protein where they do search for the modifications and they do recover the peptides. So in some cases, you can allow it as a post-translational modification and then recover it. But I think in a baculovirus system, often the glycosylations can be overwhelming and very heterogeneous. And so our approach is exactly what you said, just to start to employ some of these enzymes that will chop them off. I think that's probably the best strategy right now. And then there's more and more of these enzymes that are being published. I know David Schreimer just published one maybe a year ago that can be recombinantly expressed in your lab and used quite easily. The question is whether you want to use those enzymes before or after you do your HDX experiment, because now we have that as a possibility, right? You can say, I can do my binding experiment or whatever my, state A and state B are with all of the modifications there. And then I can just remove the modifications after quenching. Or you can decide, oh, I want to get rid of them all at the outset, and then do your state A and state B comparison. And both of those are valid approaches, of course, depending on your specific question and your specific system. But I also want to add that this sort of extra processing of samples after doing the exchange it's not uncommon. We have a lot of projects involving nucleosomes. And so we use, typical 147 base pair DNA. And over time, if we're injecting a lot of samples, this DNA will really clog up our LC system. So we just routinely run these ion exchange spin columns to get rid of it. And it's surprisingly easy to sort of incorporate these quick 30 to 1 minute steps after you've quenched your reaction without really sacrificing a lot of your deuterium signal. So that's my positive note.
That's a fantastic question, Tom. It really is something that I guess the community is really, battling at the moment, with glycoproteins becoming more and more impacting on structure and function. So thanks for that.
Yeah, I should also add that I know that there are people looking at the hydrogen-deuterium exchange of the actual modifications as well. So working out the fundamentals of sugar HDX. So that's a completely different perspective. I think we have 3 participants that have raised their hand.
Yeah, we do. So I'll just go with the way I saw them come up. So Tony, if you want to ask your question, please.
Yeah, hi, Sheena. Tony Purcell from Monash.
Hi.
And I apologise in advance. My phone rang halfway through your presentation on the coronavirus stuff. So if I missed it, you know how it is nowadays. It's a bit crazy. But I wonder if you can comment on the sorts of affinity required for the antibodies to sort of, I guess, so confidently map these epitopes, and the potential for looking at discontinuous epitopes as well, and how well you think you map those sorts of epitopes.
Yeah, that's an excellent question. So not just for antibodies, but just in general. I mean, most of the questions you just raised relate to any sort of interactions that we're looking at. So I think that the affinity question, here we had a quite tight affinity. I think we were in the nanomolar range. But in my previous example, we sort of had some of those micromolar binders. And so the affinity really only becomes an issue if you can't have whatever your molecule of interest in is in excess. So for the antibody experiment here, because we were only interested in looking at peptides from the spike protein, we actually did it, 1.25 fold. So we were really making sure we saturated the binding site, just like in the CK2 alpha, we had those fragments at, way high concentrations. And that is a step for us for every single experiment. We go to our our equations, and we put in and we estimate the percentage bound that we're going to get. In my experience, you want to always be sitting above 60 or 70% bound to see a good change. So you will see a change even if you don't have 100% bound, but it would just be much smaller. So you really, might have to dig into the statistics a little bit more. Now, in terms of discontinuous epitopes, I think as long as you have coverage across your protein, you shouldn't have any difficulty identifying them. Maybe just a little bit more challenging to interpret in the absence of structural data, right? Here we had the spike protein structure, so it was very easy to map it on. I think, did I get all components of your question?
You did. That was great. Thanks. Really enjoyed your talk. I have to head off at 11. So if you go over 11, I'll have to sneak out. But thanks.
All right. Thanks.
No problems there. Thanks, Tony. Sheena, we've got another question if you're happy to continue with the question.
Yeah, absolutely.
So we've got Robert Good, please, if you want to go ahead with your question.
Hi, Sheena. Robert Good from Monash in Tony's group, actually. I was just wondering more about the post-processing with enzymes and things. How limited are you there with having to maintain your low pH and cold, and how do you overcome that?
Yeah, absolutely. And so that really has been the challenge with these, the enzymes that remove the sugars, because there were not many of them that were really active at the low pH. So so you're absolutely limited, is what I'll say. So you want to keep it as close to pH 2.5 as possible and as cold as possible. But that being said, you of course have leeway either side. And so it really just takes a minute to optimize. So for our ion exchange column, for example, we do that at low pH. And actually that's one thing I probably wouldn't compromise a lot on, because the pH has quite an effect, but we don't do it, at super low temperatures. It's just about doing it as fast as possible and just knowing that that's going to reduce your signal a little bit, but that doesn't mean that you lose your whole signal, right? You can still detect a difference. But yes, very, very good point. You are restricted to, a low pH range and as cold a temperature as possible.
Yeah, I suppose their consistency is key to really making sure that you have the same impact throughout all your samples there. Sorry, go ahead.
I was going to say, and that's where automation can become really important, right? So if you are going to, one thing I think I say a lot about HDX is you sort of, if you ask one question with it and you set up the system, you find yourself, I can just use that same system to ask all these other questions. And so in my lab, I very rarely find that we set up a sort of a protocol and then we never use it again, right? So now we have this spike protein protocol. We can easily just pick up any antibodies that our collaborators find and, very quickly find out what their epitopes are. And I'll just jump in here in the written question and answer. Someone's asked if these extra processing steps are online or offline. And I will say they can be either. I think if they are automated, there's always an advantage for reproducibility, but they can be either. We do our ion exchange offline, but, definitely the basic step of protease digestion, that's one that we always do online. And we always try to keep it online because it's really important to be reproducible, but you can do either of them. Again, depending on your specific system.
Fantastic. Thanks for answering that question in the comments too, because I think it was definitely connected relevancy there. Yeah.
Okay, so I think if we're okay to continue, Robert, I know we're at, for me at 6 o'clock, but at 11. Yep.
Yeah, I think we're okay to continue. As I said to everyone, this is going to be recorded, so I would love everyone to stay and join us because I think this next section is going to be very exciting.
Yeah. Okay. So the next application I want to talk about involves looking at a protein interaction with nucleic acid, specifically with RNA. And this is work again by a PhD student in my lab called Naifu Zhang. So the protein of interest here is MTR4. So MTR4 is an essential ATP-dependent RNA helicase. It's highly conserved. We're working with the yeast protein, and it's absolutely required for all activity of the nuclear exosome. In other words, it takes RNA and it unwinds it, feeding it to the exosome for degradation. Now, this is the structure of MTR4. This is the apostructure, but there is several structures available, including one with RNA in the active site. It has this core domain down the bottom, which is a very typical helicase structure with this REC-A1, REC-A2 domains. This is where all of the exciting catalytic enzyme activity happens. At the other end of the protein, we have this sort of atypical domain as far as helicases are concerned, where we have this arch and this cow or FIS domain. And, you know, the crystal structures with RNA were already available when we started this project, but we really wanted to ask if HDX could tell us something about substrate preference, because it uses a lot of different RNA substrates, and perhaps about how MTR4 is regulated as it is present in various different protein complexes. So we have a lot of data on this protein, but I'm just going to pick out sort of our very initial story. And our initial story was MTR4 with this RNA substrate, tRNA IMET. This is a validated in vivo substrate. And we basically did the experiment with and without the RNA. Now, I have this side note here that our very first RNA substrate was a short DNA RNA hybrid, and this caused so many problems in the chromatography. Whenever we had it in a sample, we would just completely lose our protein signal. And I'll be honest, we never solved the problem. Not exactly sure what it is about this DNA RNA hybrid, but we never solved the problem, but we were able to move forward with this tRNA construct. So here I'm showing again these heat maps looking at MTR4 compared to MTR4 with RNA. Now, what I'm actually showing you here, and this is a long story why we have so much data, but I'm showing you 3 biological replicates. So these are 3 completely independent preps, 3 completely independent experiments, all doing the same thing. And the reason I wanted to include it here is that there's often a discussion about, oh, how reproducible is HDX data? Well, these were collected over, probably a space of 2 years. And I hope you can absolutely see, even though that we have slightly different peptides that we recover because of various reasons, I hope you can see that the pattern that we're seeing is 100% reproducible between these replicates. That's just kind of a take-home message I wanted to send, because I'm not sure why so many people question the reproducibility of HDX. But let's look at the actual result that we got. So the first thing we noticed was, wow, we have a lot of changes in this protein when we add the tRNA. How can we sort of make sense of it? Well, we noticed that we had these changes that were kind of consistent across all time points. So we kind of binned them as, one type of change. And then we had these second changes, which were much shorter lived, but much stronger. So you can see here they come and then they go. And as I go through the data, what we came to realize was that these long ones are associated with RNA binding, while these shorter ones are associated with a conformational change that occurs when the RNA binds. So I can sort of delve into this a little bit more. On the left here is showing the data for one of the time points projected on the structure. And again, it's, so consistent with what we already know about where RNA binds in this helicase core. We're able to identify, all of the regions, all of the motifs that the crystal structures, several crystal structures have shown are involved in binding the RNA. The difference, of course, was that there was never a crystal structure with tRNA. So this just tells us that the tRNA binds the same as the short RNA that was here in the crystal structure. But if we look at this sort of non-canonical arch and fis domain versus in this helicase core, now we're starting to see things that we didn't know based on the crystal structure. And the first thing that we see is that in this cow or this fis domain, that's this domain sort of sitting right here, we get these changes that very much resemble those that we see in the core. And so this led us to hypothesize that this fis domain here was in fact directly interacting with the tRNA. The second observation, as I said, was that we had these short-lived differences that were in the arch domain, and then this led to our hypothesis that this was a conformational change associated with the tRNA binding. So again, another phrase that I often say when I'm talking about HDX is, it kind of enables hypothesis-driven structural biology. Sometimes when you do the first experiment, it really just acts as a catalyst for a whole range of secondary biochemical HDX and structural approaches. Now, this idea that this arch domain was involved in a conformational change is also supported by, the variability seen in various MTR4 crystal structures, as well as in the cryo-EM. So this is pulled from the supplemental data of this paper, where you can see this is that fis domain and this is the arch, and we can see it in these two different conformations. And now we're seeing something, very similar in the solution HDX data that suggests this is a movement that happens upon RNA binding. So what did we do? There is so much data on this slide, but I guess I really just wanted to make that point of how, you can get an explosion of experiments, and also how we can detect conformational change. So on the left here, I'm showing data that we collected with 3 additional RNA substrates. And what's interesting here is that if we look at these motifs in the helicase core, so the rec domains, we can see that all of the different types of RNA cause exactly the same change. So the black is the MTR4 alone, and the other colors are with the different substrates. And you can see they all superpose exactly the same. This tells us they have the same affinity in the core region, and they're binding in the same place. However, when we look at the changes in the arm and the fist, so remember the fist we suggested was involved in RNA binding, while the arch here undergoes a conformational change, now we're seeing this graded effect, almost like a dose response, depending on which RNA we're using. So it tells us that this fist region and the associated conformational change is really dependent on which RNA substrate we are using. So this fist domain clearly acts as sort of a regulatory part of the protein in deciding how the substrate is binding. Now, to further test that this conformational change is a result of the fis domain, we did exactly the same experiments, but here we use a different MTR4 rather than a different substrate. And this MTR4 has this fis domain deleted. And again, what you can see in the core is that they all bind the same. But now in the arm, we've completely lost this graduated change, meaning that when we take away the fist binding to these RNA substrates, we now no longer see the conformational change in the arch domain. So that was a lot, but hopefully, it shows how we can detect conformational change, and also how you can manipulate both the substrate and the enzyme quite easily when you do these HDX experiments. So our overall conclusions is that the cow or the fis domain plays an important role in substrate recognition, and that the arch domain undergoes conformational change upon binding the RNA substrate. And so that's the end of that. I guess before I take questions, I just want to acknowledge my funding sources. So I had startup funds from UT Dallas, the UT Proteomics Network. I have a grant from NIGMS at NIH, and many of my undergraduates are supported by ASBMB. I don't know if I should do it now or after questions. Maybe I'll sneak it in now. I am a board member of the International Society for HDX. And so if you are doing HDX or interested in doing HDX, I strongly suggest you get involved. It's a really great group. So you can go here to sign up, and we have I think twice postponed conference planned in London in April 2022. And we are also currently about to start an online seminar series sponsored by Trajan. Thank you very much. The first one will be on April 1st. So these presentations will be a little bit more in depth than my presentation, sort of really digging into some of the more recent discoveries and details of an HDX experiment. Okay, now I've got that in there. I'll gladly take some extra questions.
Thanks, Sheena. That was fantastic. I'm sure everyone really enjoyed the presentation today, going from kinases to antibody screening and then to RNA, DNA protein binding. So yeah, I'd love to open up for questions again. Either happy to have a raise a hand question or in the Q&A. Okay, I think that one from Ash. Go ahead.
Hi, Sheena. This is Ashish from Melbourne. I was wondering if you could comment a little bit more about how did you differentiate between the binding site versus the conformational change that you observed? Because my understanding sort of has been that the conformational change would definitely appear a bit later, like in the time course studies of the HDX.
Yes. So this is definitely something that is a gray area in HDX, and by gray I mean maybe not everybody agrees. But the one thing that most people can agree on is that when you see a change in HDX, you can never assume that it's conformational or binding. Both of them are possibilities. Now, sometimes that's presented as a negative, but I present it as a positive because there are not very many techniques when you can detect conformational change. Now, in this case, I will say we may be going out on a little bit of a limb, but we've now done several projects where basically just looking at the kind of the kinetics and the shapes of the curves can allow us to bin peptides into particular patterns. Now that doesn't tell you whether it's binding or conformational, but in this case, because we had the crystal structures with the RNA bound, we absolutely knew that this was due to binding. So our idea that this was due to a conformation, we present as a hypothesis, which we then go on to test using all of these different substrates. So that's for this specific story. In other systems, what I have done is I have gone on to make point mutations in the regions that show change, with the idea that if it's a direct binding site, you would potentially expect a more drastic change in affinity than if it were a conformational change. Now, to address your comment about the timing, so there are some papers that do HDX with very, very short time points. So, you know, I'm at 10 to the 5 here. I'm well and truly, we have to check our proteins. I still folded and happy out here, but there are some HDX experiments which, you know, they're really sampling a lot of time points in this early time period. And it's those studies which have sort of made this conclusion that binding happens first and conformational change happens second. But in this time scale that I'm using, we cannot say that, because basically the secondary and tertiary structure of the protein are going to dominate more in the actual amount of deuterium that's incorporated. That was a very convoluted answer, but hopefully it answered your question.
Thank you.
Yeah.
I think we have a question in the question box, Sheena, from Anonymous. Chemical chaperones are known to protect unstructured and hydrophobic regions. How do you find these conditions challenging in HDX?
So I guess I'm not sure what you mean by chemical chaperones.
Maybe we're talking about molecules that protect regions of protein structure during, maybe during transient conformational changes.
Yeah. I guess I don't really feel equipped to comment. Firstly, because we haven't really done any protein folding work, so although you could just be talking about unstructured regions.
Perhaps they're talking about, just sort of the more transient conformational changes when maybe a protein structure is unfolded, when you have a binding of a substrate or an antibody. Can you capture those intermediates? And I'm taking a little bit of liberty on this question, so I apologize.
Okay, so I will say that, we have to remember that we're in HDX, where we're still measuring averages. So if we have proteins that are visiting multiple conformations, but that they're interchanging, so we don't have sort of, conformation 1 and conformation 2, and they're not interchanging, that we would be able to detect as two sort of separate exchange profiles. But if those conformations are interchanging, then in the HDX experiment, we're going to be measuring an average deuterium uptake. So we wouldn't be able to detect those sort of transient intermediates. I will say, I want to mention two things. If your protein is unfolding and you get aggregates, it's very easy to see that in your spectra. And so you straight away know you need to go back and alter your conditions. And the second thing, and maybe the person that asked the questions would be interested, we've done some work on an enzyme called kynureninase. And what's been novel about our HDX experiments here is that we've actually done them under steady state conditions. In other words, we're doing the HDX during turnover, and we're very careful about how much substrate we add and the time points. And our goal was really to see if we could identify not only binding of the substrate, but changes due to turnover of that substrate. And we've had some very interesting results there where we really have seen the enzyme active sites completely unfold. But this hasn't caused any problems in our experiments.
Excellent. Thanks for that extra insight. Okay, I don't think we have any more questions, or last chance for anyone that wants to raise their hand and come in on a question for Sheena. I will just comment that the second HDX conference in Banff, that is a series that Sheena just mentioned, the third one being in London. The Banff was a fantastic conference. I thoroughly enjoyed. The HDX international community is tight knit. It's small but growing, but a great inclusive community.
Yep, and very helpful.
So I think I'll leave it there. All that's left, unless you have anything else to say, Sheena, is to conclude the session. Okay, so thanks very much, Sheena. It's really been a great story that you presented across 3 different projects you've got. I really love the way that you've captured HDX's flexibility and nimbleness to be able to apply to different projects in your lab. Thanks to everyone for joining us. It's been a really great interactive presentation, and I hope that everyone else resonates with my feeling about how enjoyable this was. If you'd like to speak to us further on HDX or on how you can apply HDX in your lab, feel free to reach out to either myself or Sheena. My email address will be on the follow-up email that I send out with the live recording, so you can reach me there. I'm sure Sheena's happy for you to reach out to her as well.
Absolutely.
We'll be hosting another webinar series next month. Please join us then when Dr. Ben Walters from Genentech will be presenting one of his stories, and I think no doubt will be fantastic like this session was. All that's left to say is thank you very much, Sheena. Thank you very much, everyone, for joining us, and I hope you all have a fantastic day. Thank you very much.